All right, good afternoon, everyone. Welcome again to the Physics Department Colloquium. It is our pleasure to have today with us Professor Madhav Mani from Northwestern University. He's in the Department of Engineering Science and Applied Mathematics. He is the winner of numerous awards, only a few of which that I'll mention at the moment. So he's a Simons Foundation investigator. He is a June and Donald Brewer Chair in Northwestern. He held that from 15 to 16. A Simons Postdoctoral Fellowship, which he held, and also, very notably here, the Derek Bach Undergraduate Teaching Award from Harvard University. It's a tremendous pleasure to have our speaker today, and looking at his research interests, he has one of my favorite sentences in there. In his vision statement, he says, "Mathematics is an almost unreasonably powerful language to describe and dissect natural phenomena." So hopefully he will tell us about almost, and this language which he uses to describe all sorts of physical phenomena, including organismal development, cellular physiology, structure, and ecological dynamics. The floor is yours, Professor Mani.
Thank you. Wow. Thank you. It's actually Arnan couldn't believe this is my first time I'm at Berkeley, and I spent 4 years in California. So it's been a pleasure to be here, and I sort of am stunned by how beautiful it is. I think all of you have just normalized this. It's sort of stunningly beautiful, and it's wonderful to be here. So today, I feel like one of the joys of doing the kind of physics that I'll be talking about today is that at least a portion of it can be appreciated with your eyes. And I think reveling in that is something that one should do. And what you see here are a bunch of frog embryos that have been fertilized at the same time in a dish. And what you see are all the divisions, the different morphogenetic movements that we'll talk about. These cells are gaining different fates. They're beginning to get the different cell types and tissue layers that we're all comprised of. And eventually, if you've been watching, they of course crawl off into little tadpoles. Just something I like noticing. These two embryos failed, and they sort of died at some point during embryonic development, and they died at exactly the phase of gastrulation that part of today's talk will focus on. So something I like to emphasize, we obviously, there's a part of our sort of spirit that gets excited when you see images about the deep field and the CMB and these sort of cosmological things. Part of it is the fact that you're looking at deep time. And I just wish to remind you that when you look at something very much in the lab that you can do, here's mouse vertebra developing. There is a wave of gene expression through which every single vertebra sort of gets defined. What you're looking at is a living fossil. This thing has been evolving for billions of years, and the processes that you're observing have evolved for this incredibly long period of time, and one shouldn't forget that one is looking at that level of sort of historical, that's the kind of historical phenomena that you're looking at. But something that I like to think about, whether or not it is exactly true, I'm sure that's not quite formally true, but for me, the thing that uniquely identifies living matter is that it does function. The final state to which the dynamics of embryonic development, that they asymptote to, that they converge to, eventually you and I do things. They aren't how we were taught sort of Newtonian mechanics, which is that you have the governing equations, initial conditions, and you time integrate forward. The final product does something, and I think that makes this kind of physics very different, and it's what occupies us. Let me give a little bit of brief history into how the community of scientists have thought about embryonic development. What are some of the big pillars in play? So starting in the early 20th century, or maybe the end of the previous century, the biochemical nature of development was revealed through famous experiments where you simply took two tadpoles, you sort of cut out, you took two embryos of, say, a salamander or frog, you stitched them together, and what came out, in fact, what you did is that you transplanted a very special tissue from a certain region of this embryo, and you transplanted it into a donor, into another embryo. Therefore, that embryo had sort of two copies of that tissue, and what results is an organism that is functional, it's swimming around, it's behaving, it's doing its thing, but there are two body axes that are sort of conjoined together. And what that really brought about was the fact that there must be molecules that are being secreted and that are transported around in space and time that confer the ability to form a second body axis. This was what really gave us biochemical or molecular meaning to development. What then ensued during the rest of the century was a beautiful set of experiments and a whole sort of onslaught that eventually helped us identify the genes that really help development progress. So for example, you can take a gene, you can misexpress it or you can mutate it, and spontaneously extra copies of wings or limbs will spontaneously develop. In fact, you can take a certain gene that encodes for, say, the eye, and you can misexpress it in different parts of the body, and an eye will sort of self-assemble. So the genes that what are referred to as blueprint genes, genes that identify what should be built, not how should it be built, but just what should be here, those were identified. And of course, once you have that basis, you can begin to connect it to evolution and such. Where the field is right now is effectively trying to build these big circuit wiring diagrams. This, by the way you should try to read this wiring diagram is each of these things is a certain kind of molecule. It's a protein. The ones that are connected in red, these things actually diffuse in space and time. They make long-range gradients. The other genes respond. And these are the kind of caricatures, coarse-grained pictures of these genetic circuits that are the goal of molecular biology. That's where the field, and we're sort of trying to reveal them in more and more detail. But there's a very different kind of tradition that I have been trying to pursue, perhaps ineffectively, but where people have been trying to hint at the existence of global constraints. Not so much talk about what the organism is made of, but what are the global dynamical constraints. Some of the very early pioneers in this area were Darwin himself, who after his sort of massive breakthroughs that we're all sort of well aware of, he actually spent much of the later part of his life looking at barnacles. These are those weird mollusk-like organisms that are at the bottom of ships just above the waterline. And he would basically open them up and look at variation. He would ask, how is phenotype varying from individual to individual, appealing to the question of, in some senses, what's on the menu? If we know that genetic variation, we now know that genetic variation is random, what are the kind of phenotypes, what are the kind of states that the developed organism can live in, can express? Another version of that was put forward by Darcy Thompson, where he compared different species of fish, birds, all sorts of organisms, and said that they're actually maps, diffeomorphisms that map one organism, smooth diffeomorphisms with no bound, with no sort of discontinuities, that will smoothly map species onto each other. Again, sort of appealing to the idea that perhaps phenotypes, perhaps the structure of an organism that does function doesn't wildly vary so much. And I spent many years during my sort of first few years at Northwestern trying to study the variety of phenotypes that are expressed in the final form of the Drosophila wing. And there's this paper that has a lot of pain that went into it, that identified the existence of sort of global constraints in final form of an organism. But then my attention started to turn towards more dynamical questions. And again, this is sort of the last bit of history. Conrad Waddington has drawn this picture. This was in the sort of 50s. This is a picture maybe some of you have seen. You know, you have illustrious biology colleagues who probably show this picture. The quote is something I want to read out to you. Developmental reactions as they occur in organisms submitted to natural selection are adjusted so as to bring about one definite end result, regardless of minor variations in condition. What Waddington was trying to epitomize in this dynamical systems landscape was that if you imagine that the embryo is this marble and it's sort of working under the force of gravity, it is sort of walking down this landscape, and there's sort of very important decision points at which it sort of deviates into different valleys, but the crucial point is that these valleys are, as he referred to, canalized. Any deformation in the direction that is orthogonal to the time axis is constrained. There will be restoring forces talking more generally, that restore the system to this state that is progressing in time. And in some senses, what he's saying is that there must be dynamical constraints because the final state is very special, and we know that we all end up looking like humans regardless of the genetic and environmental variation that occurred during our development. And so the final state is robust, is reproducible, and that he conjectured that there must be such dynamical constraints. And today's talk is me trying to understand and convey to you what I think modern biological data and measurements are telling us about this picture. This simple picture of a ball rolling down a landscape, and that being a model, so to speak, of development. Can we make that more formal? Can we put some mathematics around that? What are the new classes of dynamical constraints that must be present for functioning matter? So right here, I just want to say that the group that I'm a part of, that involves people here on the left, involves people that come from physics, engineering, applied mathematics. We look at, there's always, I think, a unifying physical approach to all the problems that we look at, but we do look at a diversity of functions. We study microbial ecology, combine dynamical systems theory with machine learning to study cellular differentiation, population dynamics for tumors, and most recently, beginning to think about the design of proteins. I think that diversity of topics that we work on, for me, is necessary if I want to get at sort of any sort of deeper principles that I believe are common across sort of all functioning matter. So I like the diversity, even though I often get told I'm working on everything and nothing, which is of course not true. For me, it feels very coherent, but hopefully you see some of that today. The joys and challenges of working on this kind of physics, and yes, I do think this is physics, the joys of working on this is that even the questions haven't been asked. You really just have the phenomena, the data, and you have to sort of sit there and be the first person that can ask sort of a question that can be pursued as we would. And it sort of combines, lots of areas that I enjoy learning about. And so today I will talk about the work by Dominic Skinner and Siqi Liu. He graduated, and so this is a recent picture at his PhD graduation. And Dominic now is a postdoc at the Flatiron. So I hope you can see this. It will become clearer as the movie sort of ensues. This is the phenomena that today's talk will be about. This is the organism. It is a sea squirt that you can find in the Atlantic Ocean. Towards the end of the talk, it'll become clear why I focused on this. It is I think it just has a lot of convenient features that allow me to ask the kind of questions I want to. Part of the challenge for trying to do physics and biology is identifying systems where the kind of mathematics that we want to do, it can make progress. And here's a system that I'm focusing on now for a few years. And what you're seeing is the early embryo, which sort of starts in this movie, starts at the 4-cell stage, undergoes many cycles of division. Eventually, you even see this process of gastrulation, where it turns from a ball of cells that make a sphere into something that has a tube. It changes its topology to a torus, and we all have a tube running through all of us. And in fact, the formation of that tube is the process of gastrulation, and we'll talk about that today. I'm assuming that this is a very general audience. There must be some graduate students in the room, or some postdocs, that haven't really thought about biology in a very long time. So allow me to say simple things that they might seem simple. They're still not understood in their entirety. So let's talk about some of the simple things that are happening in this very movie. Even though I'm not sort of visualizing it, what is happening is that these cells are beginning to differentiate. The genome of this zygote, of this embryo, is switching on. Its DNA is being transcribed into RNA, and that RNA is being translated into proteins. Those proteins, one must never forget, the vision that the proteins in a cell are just a well-mixed gas is not true. They are very much patterned within every cell in space and time, and it is those very patterns that actually confer the very dynamics that you're seeing here. Those spatial temporal localizations of all these proteins allow cells to interact. So here's a cartoon of 3 cells sort of meeting at a common vertex, and the interactions of those cells are in some senses what allow all these collective modes, these collective dynamics that you're seeing here to happen. If each cell was acting by itself without communicating or interacting chemically and mechanically with its neighbors, you would not see such coherent reproducible dynamics. Of course, all of these things are sort of playing back and forth, eventually giving rise to the space-time dynamics that we observe. And today I will be talking about the mechanical and chemical patterns that you see during early development. in this organism, and the viewpoint will be very distinct. In no way do I mean it as an affront to the way most biology is done these days, which is these wiring diagrams. I'm going to try to do mere phenomenology. I will merely try to describe the mechanical and chemical dynamics in a phenomenological way. Hopefully that allows me to identify dynamical constraints. If I ever give you the impression that I have succeeded and the research program is over, that is a lie. This is still very much work in progress. Arnan will agree, this is still something we don't have consensus on. We don't have the approaches yet. But here's an attempt. For me, the starting point for the mechanical dynamics is a very simple physical observation, a key separation of timescales that I have to teach you about. This movie is a very modern movie made with a very fancy microscope called a light sheet microscope. For those optics aficionados, the point spread function, unlike confocal microscopy, is isotropic, which means you have isotropic resolution in X, Y, and Z. This is what allows you to have such beautiful movies of this process of gastrulation. You're seeing this sort of cup forming, these cells go into the sort of bulk of the, and there you see the initial sort of tube. The time scale, here you have sort of a numbers, general sense of scales. Hundreds of, tens to 100 microns. That's a good number to keep in mind for the scale of embryos, length scale of embryos. A time scale of development, that can vary. In this case, I want you to think of tens of minutes to hours. That's the time scale over which you have mechanical, geometric, morphogenetic movements. Here's an experiment you can do. You can take a low-powered microscope, focus it on one of these cell-cell contacts, which is these things that are lit up. I'm going to have to not give you a lot of details because, of course, I can't exactly tell you about how all the genetic engineering and fluorescence microscopy that goes into all of this. But just to show yourself that what you're seeing is the membranes of cells, you take a low-powered microscope, you blitz, you literally heat up that little volume. That dissociates the very proteins that are the active generators of stress. That leads to a relaxation, a mechanical relaxation in the system. And if you were to simply take time on the x-axis and measure the distance between these two fiducial markers, this vertex here and this one, and just plot that distance as a function of time, what you will see is that it's hanging out there happily. You went and disturbed it. It relaxes. The system relaxes and finds a new asymptote. This number I want you to keep in mind, order of seconds. So this crucial difference between timescales, order of seconds, which is the relaxational timescales in the system, and the tens of minutes to hours timescales associated with the slow mechanical dynamics, is a hint that what you're observing here is a system of mechanical stresses that are very close to a static equilibrium with each other. They are almost perfectly balanced with each other. That is why you have this vast separation of timescales. And for those of you that do single molecule biophysics or like to think about thermodynamics, you're very used to these kind of dynamics. What this is separation of timescales is telling you is that the dynamics that you're observing here are adiabatic. Every snapshot is in static mechanical equilibrium, and therefore the dynamics must be adiabatic, as we think in thermodynamics. What do I mean by that? When I go for a bike ride and my muscles suddenly say, I need more oxygen, of course your hemoglobin rightfully, if you're in a healthy physiological state, deliver that oxygen. At no point are your hemoglobins in anything other than one of the two native conformations that hemoglobin likes to be in, the tense state and the relaxed state. All that changes are the occupancies in your system of how many proteins are in this state versus this state. So even during this dynamical process, as I'm beginning to pant and I'm biking, at every state, all the proteins are in thermal equilibrium. The dynamics of adaptation when you're putting the load are adiabatic. So you're all very used to thinking about this way of sort of thinking about sort of biological, adaptive, dynamical systems. I'm just telling you that the same thing seems to be happening in this mechanical space. So if we are to start building a physical conjecture for what we think is happening, imagine the space of all possible geometries, if you can imagine that, of all three-dimensional aggregates of this embryo. Who knows what the axes of this space are? The conjecture is that there is some sort of lower dimensional subspace that correspond to the geometries associated with static mechanical equilibrium. And the dynamics that we're observing are adiabatic, meaning that each snapshot of the movie corresponds to an aggregate that lives on this static manifold. No dynamics off the manifold are allowed. And so if you want to now ask, okay, is this conjecture really true? Fine, you can talk about low dimensionality. How do you really mathematize this problem? Well, that involves the following steps. You first must identify the coordinates of this manifold. Then you must take real biological data, real live imaging data, fit the real data, and see whether this conjecture of adiabatic dynamics is true. And of course, you must worry about the quality of your model. And so we will talk about that at great length. Unlike a lot of people these days, I still think that a blend of sort of data-driven and axiomatic physics approaches is where one should go when analyzing biology. And so I start with a simple model. I'm going to tell you that the forces in play are very simple. There are 3 forces in play in this embryo. And this is a huge simplification. But in the sense that I hope you allow me to say, in the long time, long wavelength limit, these approximations seem to be accurate. And here they are. There is a pressure difference between every single membrane. That pressure difference corresponds to Laplace's law. The pressure gap is supported by the curvature and the product of the membrane tension. There is an effective one-dimensional Laplace law that relates the membranes that each have their sort of forces that are trying to tug the line away from its straight line. The curvature of the straight line, which is kappa, times the force along it, is trying to straighten it. The line tension along this curvilinear line is trying to straighten that line. And so you have an effective one-dimensional Laplace law here. And the conjecture of static mechanical equilibrium means that all the lines that meet at these vertices sum up to 0 in a vector-like sense. That's what the conjecture of static mechanical equilibrium is. Those equations might seem simple. They are linear. But let me tell you that those are not simple equations to solve. And the reason is you have the wrong parameterization of the problem. The curvatures, the line tensions, and the volumes of the different cells and different geometric objects are not independent from each other. So in some senses, what we must ask, this is kind of sounding like a bit of a mathematician, those equations must be solved. We wish to analytically solve them. But over what space of geometries do you want to solve that problem? And here's a key insight that we had that helps solve the problem. If the pressures are uniform, and if those are the only 3 forces in play, pressures, membrane tensions, and line tensions, and if the pressures are uniform, then the only permissible static geometries are polyhedral tessellations, things that you learned in high school, right? Simple Greek 3-dimensional objects. If you have non-uniform pressures, then a slightly more complicated geometry is permissible, what I call spherically curved polyhedral tessellations, which simply means that every membrane is a section of a sphere, and every line is a section of a circle. Now, it turns out that we have a parametrization of these geometries, and it's something you've all seen. I'm actually quite convinced you all know this. It is a generalization of the Voronoi construction. So if you remember, a Voronoi construction is you simply drop down these generator points, which are the vertices of the dual blue lattice, and then you simply say that you democratically allocate space to all those generators. You can generalize this idea by saying that every lattice point in my dual lattice has a charge. That charge, the magnitude of that charge allows that volume, that cell, to be more greedy. It makes that one more greedy and want to take up more space. And in that way, you're able to account for different sized cells. If you now gave every Voronoi generator point a color as well, that color is what induces curvatures. And that's exactly the equation for a generalized Voronoi diagram. There are 5 C numbers. So if you gave me a spherically curved polyhedral tessellation with C cells, there are 5 C numbers that you need to that I will report back to you that uniquely identify that spherically curved polyhedral tessellation. They are the 3-dimensional coordinates of your generator points, x, y, z, 3, 1 charge, 1 color. 5 numbers per cell. If you have those 5 numbers, you can account for any spherically curved polyhedral tessellation, and it's exactly over this space of geometries that we will attempt to solve the problem. Oops, I pressed the wrong button. And, oops, sorry. That's exactly the coordinates of this static manifold that initially sounded like a bit of sort of fluffy, loose thinking. The coordinates of this manifold, I conjecture, are exactly parameterized by the generalized weighted Voronoi diagram. So how well does this do? Let's now talk about how well does this actually fit the data. So you can make some synthetic data. This is what every physicist knows that you should first do. If you have a scheme that tells me I will be able to tell you these geometric parameters, you first make a synthetic data, you put noise into that synthetic data, and you see that as you increase the noise, are you able to recover the ground truth geometric parameters that you used to actually generate that synthetic data. So of course, as you increase the amount of noise, the accuracy in reconstructing the geometric parameters, of which are these 5 numbers per cell, the generalized weighted Voronoi parameters, of course they degrade as you inject noise into the system, but the degradation is very slight, and that's because the numerical scheme that we're using is a global quadratic optimization scheme that is incredibly robust to noise. And again, the key emphasis being here on global. It is not a local fit, but a global fit, which is exactly what gives these robustness, the robustness that we observe. How well does it do on real data? So here's a real embryo, a little cartoon of a real embryo. I don't know what the ground truth there is. I have no idea what parameters that embryo is right now living according to. But what I can measure is an error. You can measure the error between where the membrane pixels really are in the embryo and where your spherically curved polyhedral fit is. You can measure in a dimensionless way the deviation from the model, the geometric model, and the data. And you can simply make a probability distribution of that error. And what you find is that error is incredibly small, up in the single percentages, which means the evidence that what this is really saying is that you can account for the geometry of this embryo at every time point with a very simple model, which is 5 numbers per cell. And not only can you just plot a distribution of errors, you can actually ask where are the errors in space in the embryo. This is at a particular time point. What is the spatial map of the errors? And this project was a real labor of love. I agree that there are some places where there are errors, and in fact, those are the very cells that are about to divide. So indeed, they deviate from a spherical approximation because they're just about to sort of ingress and make a sort of new membrane. Please look at the color bar. It's not a bad error. 95% of the geometry can be accounted by such an audaciously simple model. Of course, there are deviations, but in a perturbation theory-like sense, let's talk about the first order thing that the model seems to account for. Just to give you a visual sense, I'm a very visual person. I tend to like sort of image movies like this. This is the data that comes out from the microscope where we've just segmented every cell. This is the real sort of geometry. And this is the approximated geometry using this simple model. And I hope what just comes across, it's a visual thing. Of course, here's the real data about where the errors are. But you really can recapitulate the phenomena that you're seeing using a very simple approach. But of course, we aren't just interested in talking about geometry. The model started with mechanics, forces, pressures, membrane tensions, and the such. And what I'm telling you is, by solving a very interesting inverse mechanics problem, you can go from the parameters of a generalized weighted Voronoi diagrams, which are the right-hand side of these equality signs, S, P, Q. Q is the 3-dimensional coordinate of the generator, P is the color, etc. And on the left-hand side are the mechanical quantities. We've derived these nonlinear relationships between the geometric parameters that you can infer from real data and the mechanical forces that are in play, and you can now visualize those. This is the first, I think, single-cell map in a living, unperturbed embryo of mechanical forces. This is the surface stresses on every single surface of this embryo. And this, we're not perturbing it, we're not blasting it, we're not doing anything. We're merely imaging it and then building a piece of physics that allows us to do this sort of inference. Let me teach you a little bit about this inference. I'm not going to have the time to do this in full 3D, but I would not be doing a good job if I came to this audience and didn't do just a little intuition as to where those equations come from. So let's start with a simple scenario. Obviously, the organism is not 2-dimensional, right? But I'm trying to communicate the idea where the physics comes from. So imagine you had a simple cellular system that was 2-dimensional with uniform pressures. If the pressures are uniform, what that means is that the boundaries are not curved, they're straight. And if you're in static equilibrium, it means that all the tensions that meet at these vertices are balanced with each other. That's what it means to be in static equilibrium. So you write down this little equation that simply says the vector sum of these tensions is 0. A vector sum, thought geometrically, is a triangle. It's a closed triangle. It's closed because it sums to 0. And if you think a little bit more geometrically, this triangle can straddle the vertex that it corresponds to. And if you just rotate it right, and this is not obvious, the dual triangulation, the forced triangulation, will be dual, will be orthogonal to the original cellular lattice, which means that every purple or green line in this dual lattice intersects the original black cellular lattice at 90 degrees. And that means the problem of force reconstruction in this highly simplified scenario is simple. All you need to do is jiggle around these generator points for the dual lattice so as to maximally orthogonalize all of these junctions in the dual lattice to the original lattice. It's a simple quadratic programming problem. And what you get left with is that the reconstructed dual triangulation is, you can read off the tensions from it. The length of this junction is exactly corresponding to the tension on this junction. The length of this junction corresponds to the tension on this junction. Now, how do you generalize this idea to non-uniform pressures? This reminds me of Einstein a little bit, because remember that once you have non-uniform pressures, now these tension triangulations, this tension, this triad of tensions, this triad of tensions, won't form a tessellation in space anymore, because those forces are not the only game in town. Now you've got pressure jumps across the interfaces. And so to speak, if you remember, if you take a pizza and cut out one slice of it, now there's a defect, and the only way to actually close that object is to deform into the third dimension, and you make a cone. So for that same reason, the tension triangulation here will not form a tessellation. There is a defect. If you want to think about it, there's a charge per cell that's exactly proportional to the pressure gaps that it has across its various interfaces. And there is a generalization of this construction, which is you still have tension triangulations. The tension triangulations are now orthogonal to the tangent direction along this curve, not the direction that joins the vertices, but the curvilinear direction. So this force balance relation still holds on this triangle and this triangle, but there's this weird unhappy quadrilateral that sits in the middle, and all you must use is a very simple result from multivariable calculus, which is that we have a closed compact object, and you do n ds, you just do a normal vector integral over that surface, you must get the answer 0. If you construct that integral for each one of these elementary pieces of the dual triangulation, which are these triangles, and this quadrilateral in the middle, you will exactly get the equations of force balance that you started with, which means that there is a duality, there is a bridge that exists between the geometry that you observe and the mechanics that is underpinning it. Of course, those are hidden variables, but those are the things you care about, right? I mean, of course, having a parameterization of form is nice, but that's the thing that you actually care about. And here's the result of that. Pressures, surface tensions, line tensions as a function of time. This is an early time in development, 64 cells, 76 cells, 112 cells. These are the first maps you're ever seeing of the mechanical forces in an embryo, in an unperturbed embryo, single resolution over time. And in fact, you can even begin to discern what the mechanical logic of these adiabatic dynamics are. This is a cross-section through the embryo. You're kind of cutting through it rather than just sort of seeing its south pole. And what you see is that as the cup starts to form, as gastrulation starts to occur, a mechanical bilayer seems to self-assemble with a high-pressure layer ingressing into a low-pressure layer. And you can begin to ask what is the mechanical logic of this sort of mechanical process. But just to give you some sense, you don't just get space maps, you get time maps. Okay, so here I've laid out, this is the lineage of every cell. If you just track, this is one cell at the 64-cell stage, and then it divides. Here's another one, and it divides. So this is a time map where I've heat mapped the contractile mechanical stress on top of it. And what you see are these beautiful mechanical patterns that now you can study. There is one interesting property that those of us that come from condensed matter physics worry about and think about a lot. In fact, they're the richness of some of the phenomena that we study. There are zero modes in this problem. In particular, there's a very interesting zero mode where the same geometry can be sustained by a one-parameter infinite, one-parameter family of mechanical states, where membrane and line tensions sort of trade off their contribution to the overall contractile stress. If you're interested in that, please, let's talk about that later, because in the paper, that's a big part of what we did. Let me step back and now talk about dynamical constraints. Enough about mechanics. Mechanics has ended. Now we're going to move on to sort of stepping back and connecting the mechanics to chemistry. So how do I think about this adiabatic dynamics from a dynamical systems point of view? I come from, in nonlinear dynamics, we really like to think about separation of timescales. If you have a dynamical system, and I mean this loosely, and I will try to make it formal over my career, but I mean this loosely. Imagine you just had some autonomous dynamical system that corresponded to the dynamics of gene expression being the space-time dynamics of chemistry. And then you had some equation corresponding to the mechanics. When I say there's a separation of timescale, when I say that when you go and cut one of these taut rubber bands, the mechanical relaxation is incredibly rapid, what I'm telling you is that the timescale, compared to the timescales of gene expression, there's an epsilon here, epsilon is a small number, the mechanical part of this dynamical system has much more rapid kinetics associated with it. And sort of what we always do in nonlinear dynamics is that then we go into a new time coordinate system. We introduce slow time. And when you go into slow time, what you discover is that indeed the mechanical portion, the fast portion of the dynamical system can be approximated to be steady state. That's the static equilibrium. And what that means is that you have a slow manifold. For those of you that don't like to think about this or haven't thought about this too much, let me try to give you a physical analogy. You have two subsystems that are coupled to each other. One has fast relaxational kinetics, one is slow. What that means is that imagine the system is time evolving and the slow one updates according to an Euler step, one step. The fast system updates right away. And the slow system always sees the steady state solutions of the fast system. This is what dynamical systems theorists call dimensionality reduction, because it means that a much larger possible phase space is avoided, and what you have is a slow manifold. That slow manifold is exactly conferred by the separation of timescales. That's what's saying that the dynamics will only pursue a lower dimensional space for these sort of combined dynamics. And so now I think about this picture, and I think if we're meant to think about the marble as the entire embryo, but the embryo has both chemical and mechanical dynamics, then maybe not just in this one embryo that I've studied, maybe it's a generic fact that the mechanical dynamics are fast. And what that means is that the mechanics are always going through static equilibria, and the chemistry only sees a simpler, reduced subspace of the mechanics that this embryo lives in. So that's one version of dimensionality reduction that I think is going on perhaps in development. Right, so predictions, zero mode, there's the possibility of mechanical flexibility, and in fact, as we know in condensed matter physics, zero modes are usually fluctuations that bias towards the directions of zero modes, and there's lots to talk about there. Then you should ask yourself, as a biologist, from that point of view, there must be a mechanism in place that is ensuring proximity to the static manifold. There must be some active processes that are ensuring that you always relax back quickly. I've just done mere phenomenology, but there is a strong prediction that there must be mechanochemical feedback systems that actually ensure that you're always proximal to an adiabatic manifold. And this question of whether separation of timescales is generic. Okay, so I will now move on to gene expression dynamics. There is a superb measurement that your colleagues are doing that, for those of you that don't do biophysics, I want to tell you about. It is a superb measurement. The measurement involves the central dogma. DNA transcribes to RNA, translates to amino acids fold, proteins do chemistry, they do enzymatic reactions in the cell. Here is this embryo at a 1-cell stage, 2-cell, all the way up to whatever, some stage in development. We have the ability to now, at single-cell resolution, almost single-molecule precision, not quite, make measurements of the RNA transcripts coming off from every single gene in this organism. This is called single-cell sequencing. And this has now been multiplexed to include all other sorts of sequencing technologies. But RNA is the modality I'll be talking about today. And another technology that, again, if you don't know about this, this really changes the game for trying to do physics in these embryos. They can do this in multiple embryos, and they keep track that that cell came from that embryo and that cell came from that embryo. This is called barcoding. Okay? And so that means that you don't just grind up 50 embryos and do single-cell sequencing. You grind up 50 embryos, but you remember, using a very clever genetic trick, that that cell came from that embryo, or those 64 cells came from that embryo, those 64 cells came from that embryo. There is this ability to do this now. And so the data that you get is such. Here, genes, 20,000 genes in this case, 9,000 genes in this organism. So here's a heat map, genes along the rows. I'm very bad at this. And cells are laid out along this dimension of this heat map. But I remind you that embryos, you really know that these cells belong to embryo 1, these cells belong to embryo 2, etc. Yeah, I'm giving a talk in biology on Wednesday, normalizing this data. This is something that we all know when we learn physics. When you look at this, when you're trying to solve the great cosmic distance line, you get photons from light that's out there. How do you tell distance? This is the great story of standard candles and Cepheid variables, and we figured out that by using this one kind of object in the night sky, you can figure out distances. This measurement is famously hard to put into one standard unit system. So normalization is a real place where I think physics people from the physics tradition have really contributed, and I still feel this is something that we don't fully understand. In fact, the paper that we're writing goes into some detail about this. But let me try to move you towards a many-body or a condensed matter view of gene expression. So in biology these days, they don't think about this marble as the embryo. They think about this marble as a cell. And so the natural, if you think of this marble as every cell in the embryo, then as the cell or the marble goes down this landscape, the different basins of attraction are the different cell types in your body. Heart, neurons, skin, blood, etc. But what Waddington meant, and what I think is strongly the case, is that this metaphor actually works much more beautifully if you think about this marble as the embryo, the entire collection of the embryo. And now you have the dynamics. This is in a phase space. This is the temporal dynamics from some early stage in development to some later stage in development. WT, I'm really sorry, I have cultivated, I am now part of biology culture as well, wild type. WT means wild type, not a genetic mutation. Its genome is not altered. When you alter the genome, you go into one of these alternate basins of attraction, and typically the embryo dies. So the embryonic interpretation of this picture is that one of these parts is the one that you will see during development, and when you perturb it genetically or environmentally, you can get a mutant phenotype with these alternate basins of attraction. So here's the idea of many-body dynamics. I told you that our colleagues in biology are barcoding doing single-cell sequencing, which means that you actually don't just see one marble, you see multiple marbles at these different stages of development. That's what barcoding gives you. And that means that I can not only study the average trajectory of developmental dynamics from beginning to end, you can look at the variance, you can look at the fluctuations in gene expression at any stage in development because of this ability to identify which cells come from which embryos. And now the question is, what kind of collective modes do you see? You might see no collective modes. It might just look thermal, completely uncorrelated in space. There could be embryo-wide correlations. Some embryos, for some reason, just have a higher total load of gene expression, and others have a low total load of gene expression. Or there could be coupled spatial dynamics, real coupling in space between cells that are expressing these different genes. The question is, what kind of collective modes do we observe, and can we do this? So again, I really want to emphasize this difference. I really feel if there's one aspect of the talk you walk away with, this is that slide. I once got told when I was at, I won't say which university, someone came up to me and he meant this in the most wonderful way. I took it at least in the most wonderful way. He said, oh, you're that physicist that studies the wild type. Meaning, what that means is that what I'm telling you is that you can look at just variations between different individuals that are all functional, that are all going to end up making a good embryo. They are not mutant embryos. This is the ensemble that often biologists study, which is you break something and you ask, how does it break? And from the different patterns of breaking, you try to infer what is sort of causing it, what is sort of underlying it. I'm telling you that there is a much more quantitative, nuanced thing that you can do is by merely studying the fluctuations in functional embryos, ones that make it and have babies. That's what I really mean, things that make it. That's the most sort of important takeaway. The other one is that I can't remember sort of, I guess over many decades, Jaynes wrote a collections of papers which I think were, for me, very educational. I took a lot away from them. He formulated statistical mechanics as an inference problem, using the ideas of maximum entropy and maximum caliber, said that the Boltzmann distribution is nothing other than the maximum entropy distribution, that distribution that is maximally agnostic while still getting the average energy of the system right. An Ising model is nothing but a statistical distribution that is maximally agnostic, that agrees with the first order and second order statistics of the distribution that you're trying to fit. Jaynes sort of reformulated statistical mechanics from information theoretic point of view, and these have been used by Bill Bialik and various other people in bird flocks, in neuroscience. In fact, this is one of the go-to models when now David Tank and colleagues make beautiful GCam recordings of neurons in living mice. If you want to ask what is the underlying generative probability distribution that you're observing these samples from, you use an Ising model. And that Ising model is formulated from a maximum entropy point of view. It's a beautiful way of thinking about problem and unifying a whole host of different phenomena from bird flocks to neurons, and I'm going to use it, I think, in embryos. So let me tell you how this works. You take the raw measurements. You think about them as spins. Oops. You think about them as spins. Each spin is a vector in gene expression space. Sure, you must trust me that I've done the dimensionality reduction well, but that spin is basically quantitatively encoding the state of gene expression of that one cell. But remember, this is single cell sequencing, so that cell has a different spin, that cell has a different spin, and I measure them. But the technology of barcoding means that I can measure the fluctuations in those spins from individual to individual. So maybe in one individual, it's like this, and in the other individual, it's like this. And over that ensemble of functional data, you can calculate correlations. So you identify the cell types using the first moments. This is not trivial. Come on Wednesday to my talk if you want to understand how you even discern cell types. You measure the correlations. You then impose the idea of maximum entropy, which is you simply say, there is a probability distribution rho that I'm trying to learn. I want it to have maximal entropy, be maximally uncertain, but it must match the following constraints: the means and the correlations that I observe in the data, and the biological constraints that this problem has. That is, things that I will go into, but I haven't told you that this organism, and now I'm telling you why I focused on this organism. This organism is not like humans. If I looked at 50 embryos of this organism at the same stage of development, each cell has a name and its neighbors are the same. What that means is that there is an isomorphism between the names of the cells in a different embryo, and so I can actually construct correlations across an ensemble of data because there is an isomorphism between names. And in fact, that goes one step further. The division patterns of cells in this embryo, if you took 50 embryos, the division patterns at early stages of development are the same. So the same two cells will divide this way, then they'll divide this way, then this way, and that will make muscle, and that will make neuron. These are the biological constraints that this distribution that I'm trying to infer must fit. They must respect those constraints because those are the biological constraints in the problem. For those of you that like to think a little bit about this, I just flash up. Here is the actual coupling matrix that we're going to infer. J is your usual coupling in an Ising-style model. I and J correspond to genes. P and Q correspond to cells. So the coupling matrix that you're trying to infer is the gene, it's the gene-cell coupling matrix, if that makes sense. And the ansatz that we're going to make is that within a cell, so when P and Q are the same, this coupling matrix can be dense. It can be very complicated. Genes can impinge on each other and have correlated dynamics within a cell because the genome is complicated. The ansatz that we make that the symmetric and asymmetric interactions must be sparse. They must be few. So again, dense, within cell, across cell, sparse. But we only allow the interactions that abide by the adjacency matrix that this embryo abides by, and the temporal lineage that this matrix abides by, that this embryo abides by. When you do that, you will have a distribution which will literally be Boltzmann in your usual Ising-like sense. I've forgotten the external magnetic field. The external magnetic field is fit so that you actually get the first order moments, first order statistics correct. You literally get a distribution, and I really want to tell you this is very different from how your biology colleagues think about how we should think about single cell data. They want to find, if anything, the generative distribution, the underlying probability distribution that generates the distribution of gene expression in a cell. If you sample from this distribution, you will get the gene expression state of a whole embryo. In fact, you will get it of the whole embryo in time. You get a full space-time dynamics from one pull from this statistical, from this distribution. Here's some preliminary results. Here is the stage at which we're working. It's the 32-cell stage. Here is the physical map. These are the different cell types. These are things that we found doing, I think, some of the best the sort of best statistics. Well, it's actually very much the kind of statistical physics you and I learned in school, or I guess college. This is the adjacency matrix of the 32-cell stage. This is the ancestry, the lineage, the division patterns. One cell dividing into 2, dividing into 4, dividing into 8, dividing into 16, etc. At the 16-cell stage, if you take this Ising-like model and you try to fit the data, you can fit the data. Here's the within-cell covariance, the expected, the measured covariance, and the predicted covariance. Covariance is another word for correlations. Here are the measured correlations and the ones that the model accounts for. And this model, you can see in magenta and red, is the model that allows for global modulations and accounts for sister correlations due to the division, the ancestry of every cell. And you can see that it fits the data. It gets exactly the predicted and expected agree with each other. And a simple global model can account for the across cell, the cell-cell correlation that we measure. A global model is enough. A sister model is not enough. What that means is that at this early cell stage, the variation that you're seeing, the collective degrees of freedom in gene expression that you're seeing, all the genes are high or all the genes are low. Once you get to the 32-cell stage, it's not good enough. At the 32-cell stage, here's the within-cell correlation structure. A global model fits the data, but the across-cell, the two-point correlation statistics, are not fit by a simple global or lineage model. You must account for spatial cell-cell coupling. And of course, for those of you that like to think in machine language lingo, you must agree that once you allow spatial coupling to exist, there are a hell of a lot of parameters in this statistical distribution. And so you must regularize it. You must regularize prudently. And there's a whole sort of way in which one can do this intelligently, but very sparse models seem to account for the measured correlation structure, meaning that where very few couplings in this simple embryo seem to account for the distribution of gene expression. Okay, I will stop there. I'll end with simply saying that I think a lot of us that look at biology from afar feel a little bit disappointed because all we see are these many parts that fit within a Rube Goldberg machine. Here is a gentleman, and every time he takes a sip of the soup, this automatic Rube Goldberg machine takes this napkin and wipes his mustache. And that's how we often feel when we think about these genetic circuits. But there is a different paradigm out there that I think some of us are trying to develop, which is forget about this is kind of a Wittgensteinian pivot if you come from the philosophy of language. It's saying forget what the thing is made of. Just tell me what it does. And just do mere phenomenology and try to reconstruct the dynamical constraints. Okay, a many-body effort. Sichi Liu and Dominic Skinner are these two amazing people that did the mechanics and the chemistry portion, and my collaborators at the University of Chicago and Montpellier in France. With that, thank you so much. I hope I didn't go too far.
Thank you, Madhav, for a great talk. Questions? Professor Jovanovic?
Yes, hi. So this was very interesting to me because you're answering the question of how cell differentiation occurs. But having sat through the lecture, I still don't know how cell differentiation occurs, given that they're all starting with the same DNA. Can you help us?
Good. So what I had to skip over a little bit in interest of time, let me just tell you about what is happening in every embryo, including yourself, when you were once a mere few cells. There is something called the maternal to zygotic transition. This is the earliest expression of proteins and the kind of action that happens in an embryo is not because of expression of your genome, it's because of the expression of your mother's genome. Your mother deposits RNA transcripts in the embryo, and for the first sort of epoch of embryonic development, it is your mother's DNA that is in some senses encoding and responsible for the dynamics you see. The transition from that phase to when the zygote, the embryo, truly switches on its own genome and what those first patterns are, we don't know what those are. I think that's exactly what I was trying to do here. That transition from a maternal load where what you see is this collective variation of gene expression going up and down to the spatial coupling is what I was trying to capture there. You're asking in terms of pattern formation, the way we like to think about pattern formation, how do I get the patterns I see? I don't think I still have a good, elegant answer to that question. It is still complicated.
So you brought up what is to me was going to be my second question,
Yeah.
Which has to do with epigenetics, which is the statement that the DNA is not everything. There are environmental influences that you brought up from the mother. And so is this true that DNA is not enough and you have certain environmental influences?
In fact, one of the things that Conrad Waddington did, he's famous for this idea of genetic assimilation. Genetic assimilation is the idea that an environmental stress can alter the phenotype, and when you take away that environmental stress, that phenotype stays. So in fact, there is evidence in these embryos that environment and the genome sort of come together to define what the eventual state is. Of course, in embryonic development, the egg or your mother takes incredible care in trying to get the initial conditions the same, but depending on what she ate, depending on what the temperature is, the environment does impact things.
So you're saying DNA is not everything?
Absolutely.
It's a revolutionary statement.
No, I think in embryology, the fact that environment affects things is a... I agree in the modern biological setting, maybe it sounds revolutionary, but I think if you look at the history of embryology, this is in fact, it's one of the central questions. How does environment and the genome interact to identify the final state of embryonic development? Yeah.
That was a great talk. One comment that Rube Goldberg was a Cal alum.
Oh, is it what?
Rube Goldberg graduated from Cal.
Oh, I did not know that.
So be careful if you're going to make it seem like
Did he give us money?
I'm sorry. That is the chair talking.
your discussion of the Acidian modeling of covariance at the end.
Yes.
You had 10 and 25 couplings that you added to include the description.
Yes. Yes.
Calculation, we know which cells are signaling which other cells what are signals. Could you infer what those
Good. In fact, something I skipped over, that bottom right thing, we actually get which interactions. We get a ranked list of the interactions. We get a ranked list of the interactions that are important, and indeed they agree with what we know are the FGF signals that are actually the ones that confer differential state that then allows gastrulation to ensue. So we seem to recover that. Yeah. Remember, approximately every cell in your body does have the same genome. When I was speaking of mutations, those are mutations in the first cell that then lead to a very different trajectory. So maybe I didn't, the mutations other than in cancer, the mutations are not happening on the fly during development. They happen in the original egg and sperm. Yeah, sorry if I wasn't clear about that. Good question, great question.
So the mechanics portion of the link between geometry and the forces in your terms of
Yeah.
that requires an exhaustive list of the types of forces. What are some other forces outside of the simple ones that you have consistent?
Very good question. So the forces, you saw which ones I put in, which are the ones consistent. You can ask what other forces could be in play. First of all, I think the quality of the fit forces you to say that on the time scales and length scales that you observe in that movie, an effective model must be such a simple model with just 3 mechanical ingredients. In fact, if you took a much more high resolution temporal movie, you would see all sorts of additional dynamics called blebbing on these membranes that exactly correspond to what look like more elastic-like properties. In fact, you see things like Gauss-Bonnet in the sense that an elastic sheet doesn't like to change its Gauss curvature. It only likes to bend. And you see remnants of that in the very high-speed fluctuations that you might observe. So what I want you to think about is that this model is very much effective in the way we think about in physics. Long wavelength, long time scale. Good question. Great question. Blebbing. It's called blebbing. You sort of see these little sort of little mushrooming things on the membrane.
Does your inference of forces from geometry require the assumption that basically all cells have the same stiffness tensor?
This is the difference between having a constitutive model and the principle of virtual work. The beauty of the principle of virtual work is that you need not tell me what the constitutive model is. You get a relationship between the different stresses in play. In fact, that's the only reason this is worth anything. Because if I had to instantiate a constitutive relationship, then I think that is a very shaky bridge to cross. That's very complicated. That's real material physics out of equilibrium, which we still don't have a handle on. That's a good question. This model did not have any constitutive relationships. Great question. Thank you.